Welcome back. Today I wanted to go through the phases of meiosis. On your AVQ, I had asked you to identify some drawings of meiosis, and traditionally students have a lot of trouble with this. So the whole point of this exercise is not to memorize those pictures, although you will see them on the exam. The point is to practice understanding what is happening in each phase of meiosis. So the first thing you want to do is you want to sort of overall identify the phase. So like if the chromosomes are being pulled apart, we know that's anaphase. If they're all lined up in the middle of the cell on the equator, that's going to be metaphase. If the cell's pinching together, that's the very end, that's telophase. And if the spindle fibers are growing, but the centrioles are still sort of close together, that would be more like prophase. So we can use these hints to figure out the overall phase: prophase, metaphase, anaphase, or telophase. And second, we're going to try to identify whether that would be prophase I or prophase II. I find a lot of times students will just put prophase, but that's not really telling me the difficult part. The easy part is that it's prophase. The harder part is identifying whether it's one or two. And some of the things that you can do is look and see how much DNA is there. Since we are reducing the amount of DNA in a cell, every time we do that division, the less there is, the further along it is towards meiosis II. You can look and see if their format of the chromosomes is as homologous chromosomes or as sister chromatids. And in order to figure that out, you have to look at how many arms does the chromosome have. Remember that a single chromatid looks like this. So there's just one arm. A sister chromatid format is after it's been replicated and before the sister chromatids have been pulled apart. There's two of them. And then when you have a homologous chromosome, that's when you have a similar chromosome and it's right next to it. But remember, one of these chromosomes came from the maternal component and one of them came from the paternal component. So for example, maybe this one came from the mother and this one came from the father. They're both still chromosome number 1, or whatever the chromosome number you're talking about, but they're not genetically identical. Remember, sister chromatids start out as genetically identical because we copied one chromatid from the original chromatid. All right, so let's go on. Let's look at some of these pictures. So when you look at this, the first thing you want to do is you want to decide what overall phase this is. And since we can see that the chromosomes are literally being pulled apart from each other, a part starts with A, and that should clue you in that this is going to be anaphase. But is it anaphase I? Or is it anaphase II? And these are the steps that are in each of those. So the first thing you're going to see is that the spindle fibers are going to shorten, and that's what's going to pull the chromosomes apart from each other. The centromere is going to release, so what was holding these together is going to go away, and the homologous chromosomes are going to separate in anaphase I. Whereas in anaphase II, it's very similar. Spindle fibers shorten, centromeres release, and the sister chromatids separate. So really the big difference between anaphase I and anaphase II is what's being separated. Are these in homologous chromosome format or are these in sister chromatid format? So remember, sister chromatids, let me go back one page. Sister chromatids would be just this green one here. And if you were to separate just the green one, you would have one single chromatid going one way and one single chromatid going the other way. If you look carefully at this picture, you can see that you actually have multiple arms. So there's like one arm here and one arm there. So this is an entire doubled up chromosome being pulled away. So what we're separating here are homologous chromosomes. So this is going to be anaphase I. Next one, what are we looking at? Well, this looks like telophase. We're pinching off and making two cells. We have a nuclear membrane. We don't see any spindle fibers anymore. So this is clearly going to be telophase, but again, telophase I or telophase II. And again, many of the steps are similar. The nucleus is going to reform in both of them. We're going to pinch off. These chromosomes might start to relax a little bit. In telophase I and II, you're only going to have one copy of each chromosome. So what you can see is if you look at the big chromosome, you have one copy of the big chromosome. They're different colors, but there's one copy in each, and one copy of the smaller chromosome in each of these. So in both of them, these are haploid cells, we only have one copy. But in this one, in telophase I, chromosomes are in sister chromatid format. Remember that format looks like this. And in telophase II, the chromosomes are what we call a single chromatid format, which looks like that. And if you look at these guys, they definitely have that sort of X or butterfly formation. So this one would be telophase I. We haven't yet separated the sister chromatids. Okay, this one, we have the nucleus disintegrating, we have the spindle fibers starting to be produced, the centrioles are starting to move apart, so this is clearly going to be prophase, but is it prophase I or prophase II? And as you can see, many of the steps are the same, but in prophase I, this is the interesting one, we have our homologous chromosomes synapsing, kind of snuggling up next to each other, and crossing over is going to happen. And you can see that these guys are all sort of wound around each other. And you can even see right here that crossing over has happened. A little piece of the red chromosome is now on the blue chromosome, and a little piece of the blue chromosome is on the red chromosome. So for this one, this would be prophase I. Now everybody knows metaphase because everything's lined up in the middle. Metaphase I or II. The big difference here is that, is it homologous chromosomes lining up, or is it just regular chromosomes? And you can see that these guys are sort of snuggled up next to their homologous chromosome. You have a blue chromosome snuggled up next to its homologous chromosome, which is red. You can see that crossing over has happened, because remember that happens in prophase I. So because these guys are lined up next to each other, this is going to be metaphase I. Now I did want to make a note about independent assortment. Independent assortment means that you're just as likely to get them lined up this way with blue on one side and red on the other side as the other direction. And don't forget that we're going to eventually split this cell in half, and the chromosomes on this side are going to go that way and eventually become a new cell. And these guys are going to go that way and eventually become the new nucleus. And so we are separating out the blue chromosome from the red chromosome. We're separating our homologous chromosomes. And so the way it's lined up right now, we're going to end up with chromosome 1 from the red parent and chromosome 2 from the blue parent. But we could have just as easily had these line up the opposite way, in which case you have chromosome 1 and 2. The majority of your DNA in this particular organism is from the red parent. You only have a little bit at the very end that we crossed over, so not a lot of genetic variation. You can imagine how many different ways these guys can assort if you multiply this by 23 different chromosomes. You can have half of them red, half of them blue, most of them red, none of them red, all different ways. And each chromosome assorts on its own. Okay, so this is again metaphase, and as you can see, we don't have homologous chromosomes. You can imagine what's going to happen here is these sister chromatids are going to be split apart next in anaphase. So this is metaphase II. This is just before we separate our sister chromatids in anaphase II. So again, this is telophase, and in this case, the chromosomes are not in sister chromatid format. They're actually in single chromatids, and you can see, remember the sister chromatid format looks like this, sort of the X formation, and the single chromatid just looks like that, like one single piece of DNA. That's what it looks like in G1 phase before we replicated it in S phase. So we're going back to that phase. Okay, so this one is telophase II because our chromosomes are in single chromatid format and they are ready to go, ready to become gametes. All right, next one is prophase. Prophase I or prophase II. So what we're looking at now is all the normal stuff, nucleus disintegrating, centrioles migrating, spindle fibers forming, chromosomes condensing. But in prophase I, remember we had synapsis and crossing over. We don't see that here. We see that crossing over actually already happened. And these guys aren't really like wound around each other. In fact, these are actually two totally different chromosomes. Chromosome 1 is here because it's bigger, and chromosome 2 is here because it's smaller. We only have one copy of each chromosome. So we know that we've already separated out our homologous chromosomes. So this has to be prophase II because we're not doing any of that interesting stuff. All right, anaphase. So again, spindle fibers are shortening and we are separating something. What are we separating? Are we separating homologous chromosomes? Or are we separating sister chromatids? Well, you can tell by just looking at how many arms are here. Each one of these just has one arm. And you can imagine what they look like when they get where they're going is they're going to be single chromatids. So we are separating out the sister chromatids. So this has to be anaphase II. I hope this was helpful. Please practice this. You do need to know these phases for the exam. Have a great day. See you in class.